In this video, we will study two molecular particles that can act as infectious agents, viroids and prions. Viroids are small pieces of naked RNA that can cause infectious disease in plants. Viroids lack a capsid or any form of protective coat. They migrate from one host cell to the next as naked nucleic acid and do not require a receptor to get into cells. The RNA nucleotides of the viroid do not code for any proteins. Some viroids do not cause disease. Others cause disease in economically important crops. The first viroid was identified in 1967 and was located in potato plants. Scientists have also identified viroid-like agents, which are infectious RNA molecules that cause disease in fungi. The term prion stands for proteinaceous infectious agent. Prions were first proposed in 1982 by Stanley Prusiner after studying the causative agent of scabies, a neurodegenerative disease of sheep. Our cells have a gene for the cellular PRP protein. Cellular PRP proteins are normally found in the membranes of neurons in the central nervous system. The prion PRP is the infectious form of this protein. It is a conformational isomer of the cellular PRP, simply meaning that it has a different three-dimensional structure than the normal cellular PRP. The normal cellular PRP secondary structure is dominated by alpha helices, whereas the prion PRP is dominated by beta sheets, giving it a different three-dimensional structure. The difference in three-dimensional structure changes the function of the protein. The function of the prion PRP is to enzymatically convert normal cellular PRP proteins to abnormal prion PRP proteins. Prion PRP proteins can be acquired by consumption of abnormal prion proteins, which then travel to the central nervous system, through medical procedures, or may develop as a result of mutations of the gene that produces the PRP protein. Prions are known to cause fatal neurodegenerative disorders. These disorders are referred to as transmissible spongiform encephalopathies. The term spongiform was utilized because infected brain matter has sponge-like holes that result from defects in the plasma membranes of neurons, the formation of large vacuoles, and the loss of neurons in the gray matter. There are a number of prion diseases, including Creutzfeldt-Jakob disease, bovine spongiform encephalitis, chronic wasting disease, and scabies, among many others. Currently, there are no available treatments for the diseases or the symptoms that develop as a result. All of these diseases are fatal. The three-dimensional structure of the prion PRP protein makes it more resistant to heat, UV radiation, and chemical degradation. Prions may only be eliminated by incineration or autoclaving in concentrated sodium hydroxide.